Hello everyone, welcome to today's class. Today we are going to discuss tools for recombinant DNA technology. The first important tool is the DNA manipulating enzymes, which includes which includes restriction endonuclease, DNA polymerase, and ligase. Restriction endonuclease cut or clip the DNA at specific sites known as restriction sites or recognition sites. DNA polymerase add nucleotide to the 3' hydroxy end. Then ligase, which helps in the formation of phosphodiester bond. Coming to vector are known as vehicles to transfer DNA between the living organisms or living cells. Host organisms are the organisms where DNA or recombinant DNA needs to be inserted. So we will now see one by one in detail. Now, coming to nuclease. Nuclease is an enzyme that cuts the strand of nucleic acid. Nuclease is of two types, DNase and RNase. DNase cuts DNA and RNase cuts RNA. Now, if we talk about nuclease, on the basis of actions, it is of two types, exonuclease and endonuclease. Exo comes from the word surface or out. That means it cut away from the center. That is, it cut at the terminal part. But if we talk of endonuclease, endo means within. So this enzyme cuts at the center of the recognition sequence within the DNA. It makes cut within the DNA or at the center of the DNA. Coming to restriction endonuclease are enzymes, nuclease enzyme, which cut DNA within the center. So by recognizing specific sequence known as recognition sequence or restriction sites. This stick to restriction sites and cleave the sugar phosphate backbone or cut the sugar phosphate backbone. The recognition sequence this is a recognition sequence. The recognition sequence are palindromic. Palindromic means that reads same from both the ends. So we call this palindromic. So what is restriction endonuclease? This recognizes the specific sequence known as recognition sites or restriction sites. It stick to the restriction sites and cleave the sugar phosphate backbone. Recognition sequences are palindromic, that read same from both the ends. Coming to the vector are known as carrier molecule. It helps in the transfer of genetic material. A vector must have the following properties. That means it must have a ori site. Ori means what? What is ori site? That is, a site must have origin of replication from where the replication starts. Vector must be high in copy number and cloning site. What is cloning site? The recognition sequence of restriction enzymes are known as cloning sites. The recognition sequence of restriction enzymes are known as cloning sites. A good vector has recognition sequence of many restriction enzymes, but a single recognition sequence for each are restriction endonuclease. That means cloning sites are what? Recognition sequence of restriction recognition sequence of restriction enzymes are known as cloning sites. So a good vector has cloning sites of many enzymes, has cloning sites of many enzymes, but single cloning site for each enzyme. That means what? If this is a BAMH1, HIM3, ECO-R1, PVU1, PST1. So what does it mean? Now you can see this all are recognition sequence of different restriction enzymes. So I can say that this plasmid is having the cloning sites of different restriction enzymes. But if you see BAMH1, do you find any other BAMH1? No. So here it is only specific for what? Only one type of BAM H1, not BAM H2 is not found. So here we call it what? This is known as cloning sites of many restriction enzyme but a single recognition sequence for each RE. Right? Now, coming to selectable marker. Selectable marker are used to select transformants from non-transformants. Let's understand the selectable marker with an example. This is a plasmid PBR 322. Right? Now here let me consider two sites. This site, let's say it is a gene for ampicillin resistant, and this is a site for tetracycline resistant gene. Now here in the tetracycle resistant gene, what I done? I have introduced a foreign DNA. In this tetracycle resistant gene, a foreign DNA is added. Now what will happen? Foreign DNA is added, so what we will get? We will get that our ampicillin plus foreign DNA will get a recombinant product. We get what? Recombinant vector. Right? Since it has a foreign DNA, so we will get, after combination, what we will get? We will get a recombinant vector. Now there are three situations we need to understand. Now this recombinant vector, when inserted into a cell, any kind of cell, any kind of bacterium cell, so we have three options. 1, 2, 3. The first option is what? Let me draw all the cells. Now here what is done? See, this is the new gene I have added here. Right, this is the new gene. Now here what will happen? Here the recombinants, the cell are recombinant as well as transform. So here A is a transform plus recombinant plus recombinant. Here what is happening? Cell is transformed but non-recombinant. Why we are calling it non-recombinant? Because new gene is missing. This green gene here is missing. So we called it non-recombinant because the gene which I have shown in the green color was the foreign gene, and here it is not added. So we called it transform, non-recombinant. Transform means this plasmid only is inserted but gene is not active. So we called it transform, non-recombinant. And third is non-transport. Right? Now when these three cells we grow in a presence of antibiotic, ampicillin and tetracycline, what results we get? So this is A, B and C. Now what I have done, I have started growing this three cell A, B, C. Now here what I have done, this is ampicillin in the presence of ampicillin and tetracycline. Now in the first option you see, in the first option this plasmid is already having a ampicillin resistant gene, yes or no? So that means ampicillin resistance, here also you see ampicillin resistant. In this plasmid only we have inserted this foreign DNA. So here ampicillin resistant gene is already present, yes. That's why we are writing AMPR, and in this gene we have not inserted any foreign DNA. So whenever ampicillin is present, the bacteria will grow. Why it will grow? Because ampicillin resistant DNA is present, so it will grow, right? In case of B, this one also what happened? Ampicillin resistant gene is present here only. It is transformed but non-recombinant. It means foreign gene green color is absent, but ampicillin resistant is still present, so this will still grow in the presence of ampicillin. In case of C, this one here, this plasmid is not successfully inserted, so we call it non-transformed. So here the cell will not grow because no plasmid, no ampicillin resistant gene. Coming to tetracycline, here in tetracycline, this one is the tetracycline resistant gene. Now you see in tetracycline resistant gene, I have inserted a foreign DNA. So this is foreign DNA. When I inserted the foreign DNA in tetracycline resistant segment, what happened? The tetracycline resistant gene got disturbed because I had added the foreign DNA in the same site. So tetracycline resistance got disturbed. So the A cell which carries the foreign DNA, and so we are calling it recombinant because it carries the foreign DNA. And where foreign DNA is present? In the tetracycle resistant part. So that means foreign DNA is inserted into tetracycline part. That means the sequence of tetracycline got disturbed. So in the presence of tetracycline, what will happen? In the presence of tetracycline, the cell will not grow because this resistant gene got disturbed due to addition of foreign DNA. So what has happened? It will not grow, right? Now coming to non-recombinant. Non-recombinant means what? Foreign DNA is not present here, it is absent. So if foreign DNA is absent, what will happen? This foreign DNA is absent. That means tetracycline resistant gene has no disturbance as foreign DNA is absent. So it will start growing in the presence of tetracycline because it shows resistance. Now in non-transformed cells, again, it will not grow because there is no plasmid only inserted. So it will not grow. Right. So selectable marker, we are using it to select transformed cells from the non-transformed cells or to select recombinants from the non-recombinants. Yes, that is what is the vector, is the cloning sites and the selectable marker of vector. Right? Let's see one case of plasmid PUC 8. What is PUC? Here P stands for plasmid, UC stands for University of California. Here what is happening? If we draw PUC8 plasmid, we have two genes here. One part is suppose ampicillin resistant. This part is suppose ampicillin resistant. And this part is containing a gene known as Lec-4, which produces an enzyme known as beta-galactosidase. Right? And here it also contains the cloning sites of different enzymes. EcoR1, BAMH1, right? BAMH1, etc. Now here what I have done, I inserted a foreign DNA here. This is what? This is foreign DNA inserted, right? What happened after foreign DNA inserted? Because initially the Lec-4 gene was producing beta-galactosidase, which was active. Now once I introduced a foreign DNA, Lec-4 gene got disturbed, yes or no? Lec-4 gene got disturbed? No, because already one gene was present, Lec-4. Now when I inserted the foreign DNA, the Lec-4 gene get disturbed and the function of Lec-4 gene is inactivated. So this process by which foreign DNA, after addition of foreign DNA, the normal gene gets inactivated, we call this insertional inactivation. Insertional inactivation. We call this insertional inactivation. So what is happening in insertional inactivation? We have taken a gene called beta. We have taken a gene known as Lec-4 that produces beta-galactosidase. Now normally beta-galactosidase will react with a substance known as XL. It is known as chromogenic substance. That means it gives color. And when it binds to XL, what color we get? We get normally blue color. We get blue color. But since the Lec-4 gene, this happens only when gene is active. This happens only when gene is active, right? Now, here what is happening, say suppose some reason what will happen, this beta galactosidase is not producing X. Beta galactosidase, I mean, because of insertion of foreign DNA, the Lec-4 gene which produces beta galactosidase got disturbed and it is not producing beta galactosidase. So what will happen when we grow them in the X gel? When we grow them in the X gel, what they will do? When we grow them in the X gel, they will become colorless. They will become what? Colorless. Right? Thus, we can differentiate the Lec-4 gene. We can see the activation and inactivation of Lec-4 gene by addition of foreign DNA. If that foreign DNA is inserted into Lec-4, that means it will not produce beta galactosidase. If it is not producing beta galactosidase, it does not react with the homogenic substance XL and gives colorless substance. But on the other hand, what is happening if beta galactosidase gene is produced? It reacts with XGAL and produces blue color. So here, what we can say that at last in a plate, we can see blue and white. These are blue, and let me draw the white. Now here, what does it mean? Blue color stands for non-recombinant. Non-recombinant. And white stands for recombinant. And white stands for recombinant. Right? So I hope you have got this class clear. Next class we will see the steps for recombinant DNA technology. Thank you so much, students, for joining this session.